Good morning, everybody. It's my pleasure and honor to welcome you today. I'm Maria Milan, the President and CEO of the California Institute for Regenerative Medicine, or CIRM. Thank you so much for joining this very special meeting. It's a meeting we look forward to every single year, the CIRM Bridges Meeting 2021. I first want to thank the meeting organizers at Cal State San Marcos, including the organizers Courtney Dow, Susan Kaiser, Bianca Malve, and our program director from CIRM, Kelly Shepard, who you'll be hearing from shortly. This title slide has a picture of our most recent annual reports over the past 5 years. You as the CIRM Bridges students, interns, scientists are all part of the CIRM story. So please do visit our website or have a look at the annual reports. But in the meanwhile, I'm just going to go over a very high level summary of where we are today at CIRM. I think I advanced the slides by just clicking. No. There we go. So CIRM was created because of patient advocates and because of a belief in science. And it was initially created in 2004 through a one-of-a-kind, first ever large-scale investment in science by a state called the Proposition 71, which invested $3 billion in research funding specifically for stem cell and regenerative medicine research. Since then, over 1,030 programs have been funded in education infrastructure, discovery, translational, and clinical research. The Bridges program is part of the education pillar, and we're extremely proud, and Kelly will be giving an outline or a summary of what that has yielded so far, and we're excited of what lies ahead of us in the future. CIRM funding has also funded over 70 first-in-human cell and gene therapy clinical trial, which is quite remarkable because when CIRM was first formed in 2004, we didn't even know whether we would be able to turn these amazing discoveries in stem cells into potential therapeutics, and here we are today. Just this past November, $5.5 billion has been reauthorized by the California citizens, who again supported this area of research under Prop 14. Before I go into what our plans are in the new era of CIRM, this is just a summary of what the investments have been to date. Almost over $200 million has been invested in education, which is completely linked to and intricately associated with the infrastructure programs and our research programs in discovery, translational, and clinical. What we have planned, and you'll be hearing more about this toward the end of the year, is to leverage the success of the Prop 71 era and to take this identity. The identity of CIRM is it began because of patient advocacy, and patients have always been in our center. The mission has been to accelerate treatments to patients with unmet medical needs, to accelerate the science on behalf of patients and with patients as partners. It's an accelerator through this unique funding mechanism and a hub for partnership, de-risker in that science and discoveries, which don't seem like they're yet ready for prime time, but have a lot of promise, high risk, high reward, have been funded by CIRM and has yielded over $18 billion to date of partnership with industry to take this toward commercialization. We're a funder. And based on the Prop 14 reauthorization for $5.5 billion, what we plan to do is leverage and build upon this identity to build for the next era, to promote team science for real. As you'll discover as students, as those who will enter into this field, few ideas are ever original. Science is an iterative process, but each journey, each experiment, each interpretation that each of you has in your experiences can lead to something new with these ideas. So acknowledge those who've contributed to the body of evidence, but embrace what you have experienced. Embrace the negative as well as the positive data and realize that things that may seem trivial or not really relevant today, or maybe not even accepted, could actually be the solution of tomorrow. So CIRM intends to do what it can to put structure toward promoting and rewarding team science, because we believe that will unlock the key of some of the most devastating solutions for the most devastating diseases that still exist today. One thing we've learned from the past year and a half or so is the inequities that exist in our healthcare system. That's been uncovered by the COVID pandemic, both in our ability to reach the underserved and disproportionately affected, but also to understand that as a society, we could have the technology, but we have to understand the whole person. So CIRM is meaningfully embedding diversity, equity, and inclusion into all our programs. This is an empowering thing to do, not only in our programs, but as individuals. As we look at the biology, cultures, and scientific evidence, we know that we are missing the boat if we do not realize that diversity in biology does exist, and we need to account for that early on in how we approach our problem sets. And additionally, CIRM is very committed to creating as many appropriate on-ramps as possible. Congratulations, because you are on one of the on-ramps as Bridges interns. And it is such an amazing experience for those who first took this on as an internship and then realized that they could take it on as a career in various directions. And Kelly will give you a little bit more of that. But what I want to end with is this story of Derek Rossi, who is one of our CIRM scholars. He was a recipient of one of our first educational awards when CIRM was first formed in 2004. He was trained as a molecular biologist, but decided to take a chance on this new field of stem cell biology and did a postdoc, funded initially by the CIRM Scholars Program. And if you look at his quote, by the way, Derek Rossi is a co-founder of Moderna, the mRNA vaccine that helped us get through the toughest part of the COVID pandemic. What Derek says is that he didn't set out to do this work. The mRNA project was a stem cell project, which he continued when he took on his career after his training to start his own lab at Harvard. He was pursuing mRNA as a technology for stem cell biology to get a better way to reprogram cells, adult cells, into pluripotent stem cells. But then he saw the potential for this, spun out in Moderna, and Moderna developed this technology. Just kind of wraps up the spirit of embrace every single experience you have, every experiment, everything that seems trivial in terms of a method, because that may just be the very thing that you end up pursuing later. Thank you for being part of this story. And I'm now going to turn it over to Kelly Shepard and enjoy this amazing experience, incredible day. We look forward to hearing about your experiences as interns. And we want to thank the mentors and the scientists who are helping to train and to expose the future leaders and workforce of tomorrow in this field. Thank you so much. Kelly, I will advance your slides.
Thank you, Dr. Milan, for their wonderful and comprehensive introduction. My name is Kelly Shepard, and I'm a program officer at CIRM who's been involved at one level or another with the Bridges program since its inception back in 2009. Believe it or not, this program has been active for 12 years now, and we're looking forward to having a program like this continue in the future. I just wanted to go into a little bit of the history and context of this program to bring you from the past to the present and then our bridge to the future. So if we could just go to my next slide, please. The purpose of this program has always been to create a new generation of stem cell scientists at all levels, from the technician at the bench to the future leaders and innovators in stem cell biology and regenerative medicine. There are 14 different Bridges programs around the state, of which all of you are a part. but they're all a little bit different. Some are integrated within a bachelor's program. Some of you are in master's degree granting programs, and some of you are receiving certificates in stem cell specialty or biotechnology. But what makes all of you have something in common in being a CIRM Bridges grant is there are specific activities for which you receive training. First and foremost, you're all doing hands-on research internships at host institutions around the state. Host institutions are research universities with stem cell regenerative medicine programs, or research institutes, and in some cases, even biotechnology companies. You all take an advanced stem cell techniques course to help you become proficient or at least gain experience in the culture and manipulation of pluripotent stem cells. You all participate in community outreach and patient engagement activities. These have been extremely important in the past few years and will become even more important in the future. As Dr. Milan alluded, we believe it is very important for all of our trainees in this field and alumni to have an appreciation of the perspective of patients, because that's everything we do, is eventually to help people and improve human health, and also community outreach to make sure we are reaching out and addressing the needs of unserved communities and helping become good stem cell ambassadors and explaining the value of science and how it can be of benefit to all members of our society, especially for those who are not actively able to participate or benefit from this. And we can all do better, and we will. And finally, the annual Bridges Conference, which is where we are here today, so that you can all come together, share your research, and network with one another, and talk about your accomplishments with one another and your program directors and the CIRM team throughout the year. Next slide, please. Just to give you a sense of where you're coming from, these are the location of the Bridges programs around the state. We have representation of trainees as far north as Humboldt State University and as far south as San Diego State. And hopefully these on-ramps will continue to grow and build in the future, and we can cover even more portions of the geographic diversity of our state. And finally, the next slide, please. I just want to very briefly go over a little bit of where some of the 1,600 trainees that have come through this program in the past 12 years have gone on to do. So of these 1,600, 94 of them include you, some of you who are still completing your internships and won't be actually graduating till the end of 2021. However, there are 51 of you so far. And as you can see, about 40% of you have received master's degrees, 40% are in bachelor's programs, and the remainder are in certificate granting programs. About half of you have indicated to us on your surveys that you're first generation college students. Trainees have worked with over 200 mentors over the years at over 60 host institutions. And one of the most exciting things to see in tracking this program over the past 12 years is how this number of participating institutions have grown. I just checked last night, and 45% of these hosts are biotech companies. This was 33% last year when I presented this slide. And I can tell you that this is a large increase over what this program was able to provide back in 2009 when it started. I think it's an exciting benchmark that our field of regenerative medicine is growing. It's taking the earliest steps towards a path that ends in commercialization, and more biotech companies are available to help train our their future employees and the future stem cell scientists. And finally, my last slide. Maria, can you advance, please? Thank you. Trainees from this program have contributed to about almost 300 publications over the years. About a third of the alumni choose to go on and pursue PhDs, MDs, or other professional graduate programs. And about 60% are currently employed in academic or industry research positions, about half and half in each. And here's a picture of the last time we had an in-person meeting back in 2019. Hopefully we will be able to have an in-person meeting next year, and perhaps some of you will come back as alumni, either represented on my slides or perhaps in person as a future alumni panel. And my last slide, please. Thank you again. Before we go to the scientific session, which is going to be on COVID-19, I just want to thank again, after Dr. Milan did, the organizers of this meeting, Cal State San Marcos, Susan Kaiser, Dr. Bianca Mothe, and Courtney Dow. I want to thank the program directors who are acting as moderators for our sessions. And I want to thank all of the speakers, and I want to thank all of you as well for presenting. And now, with that, I'd like to turn things over to Dr. Trevor Cardinal, who will be the moderator of our COVID-19 session. We ended last year with COVID-19. We're beginning this year with COVID-19. Thank you very much, everyone.
Thank you very much, Kelly, and welcome interns and other program directors to the first scientific session of the 2021 CIRM Bridges trainee meeting. Being the first session, I feel like I need to take a moment just on behalf of the other program directors to welcome the interns and address the elephant in the Zoom that, based on Kelly's slide that she's presented, we're not together. And that's a bummer, but we need to look at the silver linings of all that. And although the situation that we're in that didn't allow us to be together in person, at the same time produced some silver linings in teaching you all how to become research superstars or develop some superhuman powers and get the same amount of work done in the context of lab closures and partial lab closures, I think, is most interns typically get in a normal, completely accessible context. So hats off to you to that. And I think you will find in this virtual format that the organizers did a magnificent job and will address the two key goals of this trainee meeting. One, using the poster sessions to recognize your tremendous accomplishments over the last year. And two, inspiring you and motivating you as we kick you off into the next step of your professional journey. So the organizers, as I said, have done a great job getting what I would call an A-list group of researchers to present to you. It'll be kind of like a budding musician going to Lollapalooza. You all, as budding researchers, will get to hear about some of the amazing accomplishments of professionals in the field. And with that motivation, we very much hope also that you see yourself in some of their shoes. Any of the presenters that you hear from over the next 2 days could very much be you in the next couple of years. The on-ramp that Dr. Milan talked about is very much putting you on to that trajectory. And we look forward maybe to seeing you in a few years at a CIRM Bridges meeting, not necessarily on the alumni panel, but in the presenter slot. So today we're going to get started talking about COVID-19, and I will introduce both presenters. We will roll through their presentations, and then we'll have 15 minutes at the end of the talks for a Q&A, and you can ask questions through the Q&A section in the webinar. So up first, we have Yanhong Shi, who is a professor and director of the Division of Stem Cell Biology Research at the Beckman Institute at the City of Hope. Dr. Shi earned her PhD from Northwestern, did postdoctoral training at the Salk Institute before taking her position at the City of Hope. She is a fellow of the American Institute of Medical and Biological Engineering, and in her laboratory focuses on using human-induced pluripotent stem cells as both disease modelers and as therapy development tools, focusing specifically on neurodegenerative diseases and brain cancers. Very interestingly, though, we will hear today how some of the very same genes involved in neurodegenerative diseases also seem to be involved in SARS-CoV-2 infection of cells of the brain. So that will be very exciting to hear about. Our second presenter today is Dr. Karen Chrisman from UCSD, where she is a professor in bioengineering. Dr. Chrisman received her undergraduate from Northwestern, her PhD from the UCSF-Cal Joint Bioengineering Program, and did a postdoc at UCLA prior to joining the faculty at UC San Diego. Her lab focuses on the use of novel biomaterials for tissue engineering and regenerative medicine, in which she has a very strong translational focus. And she has backed up that translational focus with some entrepreneurship, being involved in starting 2 different companies, looking at the application of injectable ECM-derived hydrogels in cardiac applications, one of which is involved in a clinical trial for heart failure, which is very exciting. She's received numerous awards from the Biomedical Engineering Society, the Coulter Foundation, the American Heart Association, TERMIS, Tissue Engineering and Regenerative Medicine Society, and Society for Biomaterials. And look very much forward to seeing how this novel injectable biomaterial has found an application in complications related to COVID-19. So with that, Greg, if you can initiate the presentation, join me in welcoming these two excellent researchers to present today.
Hi, everyone. First, I would like to thank the organizer for the kind invitation. It's a great pleasure for me to share with you today our research on stem cell-based disease modeling and therapeutic development. Our research primarily focuses on induced pluripotent stem cells, or iPSCs. The breakthrough development of human iPSCs has generated tremendous interest in exploring the therapeutic potential of stem cells in regenerative medicine. Because iPSCs are pluripotent, they can be used to generate a variety of tissue-specific cells. Therefore, the promise of iPSCs is enormous. They can provide models for human diseases, platforms for drug development. Patient iPSCs may also allow cell replacement therapy with reduced immune rejection. Here is a flowchart to show how human iPSCs can be used for disease modeling. Firstly, patient somatic cells can be reprogrammed into iPSCs. Patient iPSCs can be subjected to gene editing by, for example, CRISPR-Cas9 to create isogenic controls. Both patient iPSC and isogenic controls that have the wild type genes now could be differentiated into desired cell types, then subjected to disease phenotype characterization and omics analysis to identify molecular targets. Those molecular targets can then be used to guide personalized medicine. We have used iPSCs to model a variety of human diseases. For example, we have used iPSCs to model Alexander disease, a neurodevelopmental disorder that is caused by genetic mutation in a gene called GFAP that is specifically expressed in astrocytes. Unfortunately, current rodent models could not recapitulate the key pathological features of the disease, the myelination defect. We found a solution by generating patient iPSCs from Alexander disease patient fibroblasts. We then differentiated these patient iPSCs into astrocytes, because this disease is primarily astrocytes disease, and then model the disease using patient iPSC-derived astrocytes in co-culture with oligodendral progenitor cells. We were able to detect myelination defect, the phenotype that was key in patient, but could not be recapitulated in any rodent models. Moreover, we were able to identify CHI-301, a neuroinflammatory molecule, as a key player in developing the myelination defect. In addition, we have used human iPSC-derived platform to model human CMV-induced microcephaly. The infection of human CMV is a leading cause for neurodevelopmental pathologies. However, the strict viral species specificity has excluded the use of animal models to investigate human CMV-induced neuropathology. We have used human iPSCs to generate brain organoids to provide a human cellular model for human CMV infection. Specifically, we have differentiated human iPSCs into brain organoids and subjected these brain organoids to human CMV infection. We were able to recapitulate the microcephaly-like pathology in the brain organoids. Moreover, we have used this platform to test therapeutics for human CMV. Recently, SARS-CoV-2 infection has caused the COVID-19 pandemic that has impacted many of us in different ways. Interestingly, there are about 30% of COVID-19 patients with severe symptoms has neurological manifestations. Moreover, a subset of the COVID-19 patients with severe phenotype and the neurological manifestations have viral RNA and the protein detected in the brain, suggesting that SARS-CoV-2 could directly infect brain cells. What type of brain cells could be infected by SARS-CoV-2? To answer this question, we differentiated human iPSCs into neurons, astrocytes, and brain organoids. We then subjected the neurons and astrocytes to SARS-CoV-2 treatment. We found that both neurons and astrocytes can be infected by SARS-CoV-2, as revealed by positive SARS-CoV-2 spike staining shown in red. Moreover, we were able to detect substantially elevated viral RNA level in the treated neurons and astrocytes compared to untreated controls. These results indicated that SARS-CoV-2 could infect human iPSC-derived neurons and astrocytes. In addition, when we subjected human iPSC-derived brain organoids to SARS-CoV-2 treatment, we were able to detect the SARS-CoV-2 spike positive neurons in the organoids. Moreover, when we treated human iPSC-derived brain organoids with Matrigel to induce astrocytes development, we were able to see substantially elevated SARS-CoV-2 infection in the brain organoids. Furthermore, when we co-cultured the neurons and astrocytes and subjected these co-cultures to SARS-CoV-2 treatment, we were able to detect elevated neuronal infection as revealed by increased MAP2-positive and spike-positive cells. Together, these results indicated that astrocytes could boost viral infection, SARS-CoV-2 viral infection. ApoE4 is the greatest genetic risk factor for Alzheimer's disease. There are 3 ApoE isoforms in human, with ApoE2 to be neuroprotective, ApoE3 neutral, while ApoE4 risk to Alzheimer's disease. Recently, studies have provided an association between ApoE4 genotype and the risk for positivity and severity of COVID-19 patients, with higher infection rate and more severe phenotype in COVID-19 patients with ApoE4 genotype. However, whether there is a causal link between the ApoE genotype and COVID positivity and severity remained unknown. We tried to answer this question using human iPSC-derived cells. Specifically, we performed CRISPR-Cas9 editing to change ApoE4 gene to ApoE3 to generate isogenic iPSCs with either ApoE3 or ApoE4 genotype. We then differentiated the ApoE3 and ApoE4 iPSCs into neurons and subjected these neurons to SARS-CoV-2 infection. We were able to detect elevated SARS-CoV-2 infected cells in ApoE4 neurons compared to ApoE3 neurons at both 24-hour and 72-hour post-infection, with more dramatically elevated SARS-CoV-2 infection in ApoE4 neurons at 72-hour post-infection. what is the cellular consequence of SARS-CoV-2 infection of neurons? We found out that infection of both ApoE3 and ApoE4 neurons by SARS-CoV-2 could lead to reduced neurite length and neurite number. In both ApoE3 neuron and ApoE4 neurons, with a more substantial deterioration effect in ApoE4 neurons, as reviewed by smaller p-value compared to that in ApoE3 neurons. I've shown you earlier that astrocytes could also be infected by SARS-CoV-2. Next, we asked whether astrocytes could also exhibit an ApoE isoform dependent SARS-CoV-2 infection. For that, we subjected human iPSA-derived ApoE3 and ApoE4 astrocytes to SARS-CoV-2 infection. The infection was revealed by staining for the SARS-CoV-2 spike protein showing red. We can see by quantifying the spike positive cells that more SARS-CoV-2 infection can be detected in ApoE4 neuron and astrocytes compared to ApoE3 astrocytes, similar to what we have observed in ApoE4 neurons. Moreover, we found out that the ApoE4 astrocytes exhibit more severe response when subjected to SARS-CoV-2 infection, with enlarged astrocyte cellular body mimicking reactive astrocytes in ApoE4 astrocytes compared to ApoE3 astrocytes. The ApoE4 astrocytes also exhibit elevated percentage of fragmented nuclei, an indication of apoptosis. These results indicated that ApoE4 astrocytes are more prone to SARS-CoV-2 infection with severe cellular phenotype. Next, we asked what could have caused the higher infection rate in ApoE4 astrocytes compared to ApoE3 astrocytes. For that, we examined the expression of host factors important for SARS-CoV-2 infection. We found that factors involved in endosomal entry, for example, ACTR2, was elevated with elevated expression in ApoE4 astrocytes compared to ApoE3 astrocytes, suggesting that increased endosomal entry could contribute to higher infection rate in ApoE4 astrocytes compared to ApoE3 astrocytes. Next, we asked whether human iPSA-derived neurons and astrocytes could serve as a platform to test antiviral treatment. For that, we used remdesivir, an antiviral drug that has been granted emergency usage for SARS-CoV-2 infection by the FDA. We treated both neurons and astrocytes that were subjected to SARS-CoV-2 treatment with remdesivir. We were able to see substantial inhibition of viral infection in both neurons and astrocytes in SARS-CoV-2 infected cells. Moreover, treatment with remdesivir could rescue SARS-CoV-2 induced neurite degeneration in neurons substantially. In addition, treatment with remdesivir could also prevent SARS-CoV-2 induced cellular apoptosis in ApoE4 astrocytes. In summary, I have shown you that SARS-CoV-2 could infect human iPSA-derived neurons and astrocytes. ApoE4 neurons and astrocytes are more prone to SARS-CoV-2 infection. Furthermore, we show that ApoE4 astrocytes exhibit severe cellular response upon SARS-CoV-2 infection, with enlarged cellular body mimicking reactive astrocytes and more cellular death. Moreover, we demonstrated that human iPSC-derived astrocytes and neurons could serve as a platform to test antiviral drug. For example, we showed that remdesivir could inhibit SARS-CoV-2 infection in both neurons and astrocytes and rescue the neurological phenotypes. In addition to modeling SARS-CoV-2 infection using human iPSC-derived cells, we could also develop a cell therapy using human iPSCs. For example, depletion of functional NK cell have been detected in COVID-19 patients with severe phenotypes. We are now developing a human iPSC cell therapy for COVID-19 by differentiating human iPSCs into hematopoietic stem progenitor cells, and then further differentiate those cells into NK cells. Next, these NK cells will be tested for their effect to eliminate SARS-CoV-2 infected cells. With that, I'd like to thank our lab members who have contributed to the study. The COVID-19 modeling was led by postdoctoral fellows Chen and Mingzi, while the COVID-19 cell therapy study was led by a postdoctoral fellow Yue. The AXD modeling was led by graduate student Lily, who is now a postdoctoral fellow at Stanford, while the CMB modeling was led by staff scientist George San. I wanted to thank Vasey from UCLA for his support of our COVID-19 study. And a special thanks to CIRM, who has provided a generous support for several of our projects, including disease modeling of Alexander disease, cell therapy development for Canavan disease, and the recent DISC2 award for developing cellular therapy for COVID-19. Thank you all for your attention. I look forward to our discussion in the Q&A to answer any of your questions.
Hello, everyone. So today I'm going to be talking about some of our work on using injectable pro-healing biomaterials to treat inflammation associated with severe COVID-19 pathology. So first, just my disclosures. I am co-founder of two companies, one of which does work with extracellular matrix technologies that I'll talk about today, although the company is focused on cardiovascular disease. So first, before I get started, just give a brief overview of what my lab works on. So we work on injectable biomaterials for treating a variety of conditions, but mainly for basically tissue repair and tissue regeneration. So historically, we focused on using these injectable biomaterials to treat myocardial infarction, peripheral artery disease, so two cardiovascular diseases. Also, the past few years, we've been going into pelvic floor disorders, so in women's health applications. And then today, I'll talk about one of kind of our offshoot projects that was really spurred on because of the pandemic that we've been all living through. So my lab uses a variety of different biomaterials, extracellular matrix or naturally derived, which is what I'm going to focus on today. Although we do work with synthetic as well as nanoparticles, synthetic polymeric nanoparticles. And in all cases, we really try to design minimally invasively deliverable materials. So injectable materials that in some cases, like for the heart, can be delivered with catheter. And then in other cases, like I'm going to talk about today, as simple as intravenous delivery. So really to avoid surgical applications, which increase patient risk and recovery time. So one of the technologies that my lab initially developed were extracellular matrix hydrogels, particularly those derived from muscle. So we started with cardiac and skeletal muscle. And so we designed these materials as a platform or a new microenvironment to stimulate healing. So with regenerative medicine, you often hear about cell therapy to stimulate and repair a tissue, but we take the opposite approach, which is using the extracellular matrix of a tissue to stimulate endogenous cells to come in, migrate into a material, and essentially create a healing wound inside of a damaged tissue. And so the way we create this biomaterials, we use what nature has given us to begin with, which is porcine muscle. So we use pigs because they're a readily available source. There's a lot of actually porcine-derived medical products that are on the market. So we take porcine muscle. You can either, actually you can do this from other tissues too. We've done this for many tissues, but our standard is either taking cardiac or skeletal muscle. We stir it around in a detergent, SDS, to remove all the cells, or what's called decellularization. And then we lyophilize it and mill it into a fine powder and use an enzyme, pepsin, to partially enzymatically digest it. And so what happens is we are destroying that original three-dimensional structure, which is important, but we gain injectability by having this liquid form. And then when you inject it back into tissue, it does reassemble into a structure that's actually quite similar to the original extracellular matrix. So here you can see this is an injection of our myocardial extracellular matrix hydrogel into a rat heart. And you can see the arrow is pointing to how it's reassembled into a porous and fibrous scaffolds. That's actually quite similar in terms of pore size and fiber diameter to the original extracellular matrix. So this step here, before we've done further processing, is what this inset is showing here. So you can see the structure is quite similar. And then if you look at it with a scanning electron microscope, you can see it has this nice nanofibrous architecture, very similar to the original extracellular matrix. So we've used this in a variety of preclinical animal and really shown that you get tissue repair and regeneration. So this is showing an example of improvements in cardiac function. Blue here was our myocardial matrix hydrogel delivered 2 weeks after giving, in this case, this data is from a pig model, a myocardial infarction. We've also tested our skeletal muscle extracellular matrix hydrogel in a hindlimb ischemia model as a model of peripheral artery disease. And you can see in red here is just saline. This is looking at percent perfusion, so a surrogate for blood flow. Saline will plateau, so rodents will naturally regenerate, which is why you see the slope going up, but saline will plateau. Whereas we injected 2 different extracellular matrix hydrogels, a skeletal muscle, as well as actually one derived from umbilical cord. And both extracellular matrix hydrogels were able to stimulate this wound healing response and able to stimulate increases in vascularization and perfusion. But we found that it's actually, in terms of stimulating kind of more tissue repair and more appropriate muscle regeneration, particularly we think it's driven mainly by skeletal muscle stem cells, which can be labeled by PAX7. We saw significant increases with our skeletal muscle extracellular matrix hydrogel. So we've really seen that this, by injecting a new microenvironment, by replacing kind of a diseased or injured microenvironment with a more healthy extracellular matrix cues, you can stimulate healing wounds inside of these diseased organs and stimulate tissue repair and regeneration. So overall, for just a brief summary for about more than a decade of work, overall we've shown that extracellular matrix hydrogels can be pro-survival, they recruit stem cells, and they're immunomodulatory. So that's kind of a key point for the rest of what I'm going to talk about, mainly related to trying to treat COVID, is that we see with these hydrogels, we promote a pro-remodeling versus a pro-inflammatory response. So for those of you who have learned some immunology, more of a Th2 T cell type response and an M2 macrophage response, which promote more tissue healing. The cardiac extracellular matrix, just to show you there is potential for clinical translation with this. We have completed, or the company I co-founded, Ventrix, has completed a phase 1 clinical trial in myocardial infarction patients. And this is published if you're interested in learning more. But just wanted to bring up briefly today to show you that you can take a porcine-derived extracellular matrix hydrogel and translate it into patients. So that was our original extracellular matrix hydrogel technology that we work a lot with in my lab. But we've recently developed a new formulation that instead of delivering it directly in tissue, you can actually deliver it systemically, meaning through the bloodstream. So what we do is we take that same processing that I showed you to create this liquid version. But if you notice, this liquid version isn't soluble. It's actually a translucent suspension. So what we did is we did a high-speed centrifugation protocol to isolate out the soluble component as well as the very, very small nanoparticulate, and got rid of these larger submicron particulate that you would see in our normal formulation. You can also re-lyophilize that or dry it, so you can add just sterile water when you're ready to go. So it's a kind of an off-the-shelf available material. And then if you inject it into tissue, it'll still gel. But what was unique about this formulation is that when we delivered it systemically, so into a blood vessel in the case of a myocardial infarction model, or even just through intravenous infusion, we got targeting to the areas of leaky vasculature where there's inflammation. So this is showing you in a rat myocardial infarction model. We tagged the material with fluorescent dye, and you can see here it's localizing to the region of an acute myocardial infarction where you have leaky vasculature and you have inflammation. And what we found is that the material targets this leaky vasculature and then appears to gel within the gaps and partially seal off that leaky vasculature. So if you inject this into an animal that's a healthy animal, you see no targeting to any of the vasculature. And the injections we found we've already known that this material actually was hemocompatible. Our original version of the hydrogel was hemocompatible. Little counterintuitive, for those of you know, extracellular matrix, normally if it's exposed in the body, clots. However, we think that because of our concentration and the digestion processes we render this hemocompatible. So this new formulation now, if you inject it systemically, actually seems to target this leaky vasculature. So now we have this new formulation, and I will say in a myocardial infarction model, it was able to promote myocardial salvage and improve cardiac function. So we now have this new infusible extracellular matrix that can be delivered intravascularly, so IV, intravenous infusion. We found in the MI, the myocardial infarction model, that it reduces vascular leakage. So in areas of leaky vasculature and inflammation, you get leakage from the circulation from the blood into the tissue, which can create all kinds of damage. We found that, like our original hydrogels, it is immunomodulator, stimulating a pro-remodeling versus a pro-inflammatory, so really anti-inflammatory pro-remodeling response. We also found that was pro-survival, which we'd seen with our 4 normal formulations. And so because of this mechanism of reducing vascular leakage at sites of inflammation, mean immunomodulatory pro-survival, we thought now the systemic delivered version may have the potential to treat other injuries and disease with leaky vasculature and inflammation. And so something that has been dominating our lives for the past more than a year is obviously COVID-19. So I think many people are aware now that COVID is not, most patients are, when they die of severe COVID, it's because of a respiratory failure and acute respiratory distress syndrome, basically a severe pneumonia. But I think everybody's probably aware now it's affecting many other organs and really basically causing this hyperactive inflammatory response. It doesn't seem that it's actually the virus that's, killing these patients. It's this hyperactive inflammatory response. And there were suggestions that there was leaky vasculature in all of these areas where basically COVID is doing damage, but particularly the lungs, which seems to be the most severe issue. And so we hypothesized that we could deliver this new infusible extracellular matrix, target the areas of intense inflammation where there would be leaky vasculature, target the material to that, and then be basically anti-inflammatory and pro-remodeling. So to try to, we're not going to treat the infection, but try to dampen the overactive inflammatory response. So we used a surrogate model, which is called a lipopolysaccharide model. You inject LPS, which is from bacteria, into a mouse model, intraperitoneal, so IP injection. We did actually 2 injections to create really severe inflammation, but particularly in the lungs. And then we looked to see if we could get retention, in this case, in the lungs, which it does, the LPS does a lot of damage to the lungs. And so this is really a model of severe inflammation. So not a viral infection, but severe inflammation. So as a model of severe COVID pathology. And this is typically a model also for sepsis and acute respiratory distress syndrome. So we found when we infuse the material in healthy animals, you get very little retention. In the LPS injected animals, we got really nice retention and localization to the lungs, but we also saw it in the other organs to a lesser extent, since they're also inflammatory. We then repeated this again with comparing injections of IESIM to saline, looking at multiple organs and looking at gene expression using a nanostring immunology panel, and then a multiplex ELISA to look at cytokine levels, so actually protein levels. And so we found across the board in multiple organs, so lung, heart, brain, and kidney, that we looked at, we really got down regulation of inflammatory pathways, inflammatory genes, and cytokines. And then we either got upregulation or partial upregulation of adaptive immunity, the alternative activation of the immune response, and defense response. So that really does suggest, it's very similar to what we've seen in these other kind of tissue regeneration models, that it's anti-inflammatory and pro-remodeling, which would hopefully be advantageous for dampening the immune response, but then also activating the adaptive immunity to try to combat an infectious agent without doing significant damage to tissues. And then for the protein level, so looking at cytokines, we saw consistent downregulation or consistent decreases in IL-6, which is one of the main inflammatory cytokines, but particularly in the lungs, which is what I said LPS does, a lot of damage to. We got downregulation of many of the cytokines and also saw significant decreases in cytokines in the plasma, so in the blood. Really, again, confirming that it does seem like by using this infusible extracellular matrix, we can dampen inflammation. So with that, just briefly, I've shown you how we have this new infusible extracellular matrix that we think can treat multi-organ inflammation. It's a brand new delivery modality for decellularized extracellular matrix, which has been typically just implanted or injected into tissue previously. And so now we have the systemically deliverable version of an immunomodulatory biomaterial that we think has potential to treat cases, with a vaccine. Hopefully there's going to be less and less cases worldwide of severe COVID-19, but we think this could have potential for still treating the existing cases, but also other infectious agents that may cause ARDS, the acute respiratory distress syndrome, or even potentially sepsis. So with that, I'd like to thank the great members of my lab, both current and former, who've really done a lot of the work with the extracellular matrix hydrogels, particularly Ray Wang and Ann Lyons, as well as Ryan Middleton, who worked on the COVID-19 project, as well as our collaborator who's been in the ICU treating COVID-19 patients this entire pandemic, is Mark Hepikoski. And a special thanks to CIRM, who funded this special COVID-19 grant. So thank you very much, and I'll be happy to answer questions. Thanks.
Okay, thank you very much to two great presenters. There's a number of questions in the Q&A section, and we have about 6 minutes or so to address those. So I'm going to encourage us to jump right in. So the first question that is still open, I believe, is for Dr. Shi. And the question is whether because randomizir prevents neurons from being infected, what do you think about people who say that neurons are not infectable at all?
Can you hear me? Basically, people can, like both RNA and the protein can be detected in the brain. So this suggests that the virus can go to the brain and infect. The reality is that it's difficult to detect it because mostly we are trying to detect the viral infection in postmortem brain tissue. And we know that after infection, even in a tissue culture dish, the viral level will keep decreasing along the course until undetectable. So therefore, we have difficulty in detecting robustly the viral RNA and the protein. I think that is the beauty of our human iPSA model system. Actually, it allows us to track the viral infection from the beginning to the end, so that we can say, the level goes up rapidly. After 24 hours, we can see RNA level very rapidly, and the protein level becomes very high at 48 hours. 72 hours, the protein level becomes decreased, and 96 hours is barely detectable. So I think that we cannot detect much in the patient brain doesn't mean the virus were not there. So at least in the dish, we can say very clearly the virus can infect both neurons and astrocytes, and they have the neurological phenotypes.
Great, that makes sense. The next question, again, for Dr. Shi, is, do you have a hypothesis as to how the ApoE gene product affects the SARS-CoV-2 infectivity and severity?
Yes, for the infectivity, what we hypothesize is that the ApoE protein can be involved in the entry. For example, the endosomal entry, because we have shown that in ApoE4 astrocytes, the expression of host factors such as ACE2, ATCR2 can be elevated, and those are involved in the endosomal entry. Thank you.
Cool, thank you. And then I'm going to switch up a little bit the order to get some questions of both presenters. So for Dr. Chrisman, apologize if this was missed, but have you characterized your ECM hydrogels, and do you know the possible mechanism through which they are inducing their immunomodulatory effects?
Yeah, so I didn't mention it, but we have published in several papers different characterizations. So we've done a pretty thorough characterization of both the biochemical composition using ECM targeted mass spec in collaboration with Kirk Hansen, who's an expert in that. And then also physical properties of the hydrogel. And in terms of their effects for immunomodulatory, so we think it's very similar to what kind of the field of decellularized extracellular matrix has shown in general, which is that it contains, some of which are, you have damps that are exposed. There's also a suggestion there may be some remnants of basically extracellular vesicles that are in the matrix too, so that it's multi-component. So it's really hard to pinpoint, like say exactly one component, but there are studies in literature with decellularized ECM that show kind of multiple of these components can help drive particularly like the macrophage response as well as the T cell response.
Cool, thank you. And then another question for Dr. Chrisman: how do the hydrogels maintain their mechanical strength in the defect site without any specific cross-linking? And a secondary question: have you seen any impact on the kidneys or blood pressure after the IV injection?
Yeah, so the hydrogels are actually very weak. So for those who know, when we do rheometry, the storage modulus or stiffness is about 10 Pascal. And the stiffness of, say, myocardium or muscle is 10 kilopascal. So it's very, very weak. So it's not providing any type of mechanical support, but it is still able to gel and create that fibrous network inside of the tissue. So it basically is, it's cross-linked through secondary interaction. So it's a self-assembly similar to a collagen gel. A pure collagen gel will assemble through secondary interaction. So it provides that. So it's not a covalent cross-linking, but it is cross-linking in some sense. And then effect on kidneys. We've looked at measures just for like doing blood chemistry panels and looking at different levels in the blood to see if we're affecting systemic, potentially systemic organ failure, which we would not want to do. We have not seen any indication of that. We haven't measured blood pressure, which is a good question though. But so far we haven't seen any, systemic or other organ toxicity. And we've also done pathology too, looking at tissue seconds, including the kidneys of different organs. So far it suggests that everything's fine, but we still need to do additional safety evaluations.
Excellent. Thank you. Well, it's 11 o'clock, and I'm going to do my job as attempting to be a good session chair and bring the question and answer session to a close. We have a 15-minute break before the next poster session. There still are some unanswered questions in the Q&A box, and I'll just make the request of Dr. Chrisman and Dr. Shi. If you see some questions in there that we weren't able to address, if you can just click on the type answer button and provide a response during this break, we greatly appreciate it. And for everybody else, if you don't have a question, you can use this time to get yourself some water, use the restroom, whatever you need to do, stretch your legs, and then we'll be diving into the first poster session for posters 1 through 31 at 11:15. Thank you, presenters.
Thank you.
Yeah, thank you.